Welcome back to our continuation of main topic lipids. Today we will be discussing about metabolism of carbohydrate to cholesterol. As we have discussed in the previous lecture of cholesterol, about the biosynthesis of cholesterol, let's take a preview and start from there. Cholesterol is normally available in the diet, but it can also be synthesized from acetyl-CoA derived from glucose. Cholesterol is made in the liver from glucose via the pentose phosphate pathway, which generates NADPH and glycolysis, which produces acetyl-CoA. Acetyl-CoA is then metabolized to 3-hydroxy-3-methyl-gluteryl-CoA, HMG-CoA, which is reduced by NADPH in the presence of HMG-CoA Reductase, the regulatory enzyme for cholesterol synthesis, to form mevalonate. Mevalonate is then metabolized via more than 24 intermediates to form cholesterol. Now let's move ahead and learn about the HMG-CoA Reductase and how it regulates cholesterol biosynthesis. HMG-CoA Reductase is the major control point for cholesterol biosynthesis and is subject to different kinds of metabolic control. This HMG-CoA enzyme is used for a couple of pathways. If it is in the cytosol, then it undergoes a process by an enzyme HMG-CoA Reductase and produce mevalonic acid. Here, please note that HMG-CoA Reductase is an extremely important enzyme in cholesterol synthesis pathway. It is a rate-limiting step. Moving on to HMG-CoA Reductase, its synthesis is actually induced by the transcription factor, steroid regularity element binding protein, or SREBP. Before we move further, let me introduce you the important short forms that are going to be used in this cycle. HMG-CoA stands for hydroxymethylgluteryl coenzyme A. SRE stands for sterol regulatory element. SREBP stands for SRE binding protein. SCAP stands for SREBP cleavage activating protein. AMPK stands for adenosine monophosphate activated protein kinase. INSIG stands for insulin-induced gene protein. After knowing all these terms, let's see how HMG-CoA reductase play its role. First role is sterol-dependent regulation of gene expression. Expression of the gene for HMG-CoA reductase is controlled by the transacting factor, sterol regulatory element binding protein 2, or SREBP2, which binds DNA at the cis-acting sterol regulatory element, or SRE, upstream of the reductase gene. Inactive SREBP2 is an integral protein of the SER membrane and associates with a second SER membrane protein, SREBP cleavage activating protein, or SCAP. When sterol levels in the SER are low, the SREBP2 SCAP complex moves from the endoplasmic reticulum to the Golgi. In the Golgi membrane, SREBP2 is sequentially acted upon by two proteases, which generate a soluble fragment that enters the nucleus, binds the SRE, and functions as a transcription factor. This results in increased synthesis of HMG-CoA reductase and therefore increased cholesterol synthesis. However, if sterols are abundant, they bind SCAP at its sterol sensing domain and induce the binding of SCAP to yet other ER membrane proteins, the insulin-induced gene proteins, or INSIGS. This results in the retention of the SCAP-SREBP complex in the SER, thereby preventing the activation of SREBP2 and leading to downregulation of cholesterol synthesis. Next role is sterol-independent phosphorylation, or dephosphorylation. HMG-CoA reductase activity is controlled covalently through the actions of adenosine monophosphate, AMP-activated protein kinase, AMPK, and a phosphoprotein phosphatase. The phosphorylated form of the enzyme is inactive, whereas the dephosphorylated form is active. Important thing you remember here is that cholesterol synthesis is decreased when ATP availability is decreased. It is because AMPK is activated by AMP. Third role is hormonal regulation. The activity of HMG-CoA reductase is controlled hormonally. An increase in insulin favors dephosphorylation, activation of the reductase, whereas an increase in glucagon and epinephrine and elevated levels of cholesterol has the opposite effect. Now move forward and learn the pharmacological treatment of hypercholesterolemia using statins. Statins are also known as HMG-CoA reductase inhibitors. It is a class of lipid-lowering medications that reduce illness and mortality in those who are at high risk of cardiovascular disease. They are the most common cholesterol-lowering drugs. Despite of its great use, it also has adverse effects. The best recognized adverse effects of statins include muscle pain, fatigue, weakness, and rhabdomyolysis, where rhabdomyolysis is a life-threatening condition caused by muscle breakdown and muscle death. The statins are reversible inhibitors of HMG-CoA reductase and inhibit cholesterol biosynthesis. Statins are structural analogs of HMG-CoA and are reversible competitive inhibitors of HMG-CoA reductase. It is used to decrease plasma cholesterol levels in patients with hypercholesterolemia. The resulting fall in cellular cholesterol concentration increases expression of low-density lipoprotein or LDL receptors. Therefore, more LDL cholesterol is removed from the blood. By lowering blood concentrations of LDL cholesterol, statins have made a dramatic impact on the prevention of cardiovascular disease. Please note, as statins restrict the formation of mevalonate and consequently the formation of all other downstream intermediates involved in cholesterol biosynthesis, also be restricted. Statins drugs include atorvastatin, fluvastatin, lovastatin, pravastatin, rosuvastatin, and simvastatin. If we go a bit deeper, you will see that structure of HMG-CoA and statins have structural resemblance. Portions of statins clearly resemble HMG-CoA. However, the bulky hydrophobic groups of the inhibitors differ from CoA moiety of the substrate. Next, we go further. You would be surprised to know that byproducts of mevalonate are also involved in respiratory chain. The production of farnesyl pyrophosphate, FPP, and its product ubiquinone will be decreased, since ubiquinone is an essential component of the respiratory chain, which is needed for a TP biosynthesis. It is possible that the statins could compromise the ATP production needed for energy metabolism in exercising muscle. This could be responsible for the muscle cramps or weakness experienced by some patients treated with statins, and it has been suggested these patients might benefit from supplementation with ubiquinone. It has been mentioned earlier that ubiquinone is an important byproduct of cholesterol biosynthesis. However, note that other byproducts are dolichol and vitamin D. Now let's see how cholesterol is being transported from the liver to peripheral tissues. Once cholesterol has been made in the liver, it must be transported to the periphery where it is needed. However, since it is not soluble in the aqueous environment of the blood, it must be packaged in very low-density lipoproteins, or VLDLs, for transport to the tissues. Whereas, dietary cholesterol is similarly transported from the gut in chylomicrons. On the other hand, there is a reverse transport, which means transport of cholesterol from peripheral tissues to the liver. Cholesterol is removed from peripheral tissues by high-density lipoproteins, or HDL, which are frequently praised as being good lipoproteins. That's all for today's topic. More videos like this, keep watching sqadia.com.